The design of novel antimalarial drugs in the field of medicinal chemistry is an ongoing and challenging process. Production of potent antimalarial drugs that are both cost-effective and non-toxic to humans is quite a challenge. In addition to that, there's increasing resistance to the currently available therapies against malaria. Associate Professor Luke Garrett from the School of Biochemistry and Molecular Biosciences and fellow researchers from the Institute of Organic Chemistry and Biochemistry, Academy of Sciences of the Czech Republic have led research into development of novel antimalarial drugs, namely N-branched acyclic nucleoside phosphonates. The basic structure of the N-branched or ASA ANPs can be synthesized using chemical reactions, namely the ASA-Michael addition or the Mitsunobu reaction. These reactions and the use of different reagents using mechanisms such as nucleophilic substitutions, additions, and eliminations enable the nitrogen to be placed at different positions and different side groups to be attached. The different positions of the nitrogen atom and the different side groups affect the orientation and the structure of the molecule in 3D space, which in turn affects the way it reacts with the enzyme. This is important in biochemistry because the orientation of the molecule in space affects the efficacy of the drug. In this study, different such drugs were synthesized and tested for potency and selectivity. These drugs were also tested for toxicity in mouse and human cancer cells, and no significant levels of toxicity were measured. The N-branched acyclic nucleoside phosphonates selectively inhibit key enzymes in the production of nucleotides that are the basic constituents of the DNA. These enzymes are present in humans as well as different species of the malarial parasite Plasmodium. But unlike humans who can acquire such nucleotides from nutrition, the parasite Plasmodium produces its own nucleotides and is thus highly dependent on the activity of this phosphoribosyltransferase transferase enzyme for its survival and reproduction. These novel antimalarial drugs with different modifications in the structure have led to the production of very potent inhibitors that selectively attack the enzymes of the malarial parasite Plasmodium alone and not the humans.